There we go. 40 up.
All right.
Okay, Ian,
so
Ian, if I can just ask you a quick question. The whole premise, obviously, is that that oxygen event should have actually destroyed all of those unicellular forms of life.
correct. That's
okay,
the conventional wisdom.
Okay, but it didn't happen like that. So go ahead.
All right. So what we're saying is that although oxygen is vital to most visible forms of life, it's still lethal in the absence of suitable defenses, which it is assumed existing anaerobic life did not have. Therefore, the prevailing orthodoxy says that the rise in oxygen levels led to this anaerobic holocaust. An absolute cataclysm for the essentially universal anaerobic forms of life at that stage. And here's evolutionary biologist Lynn Margulis on the subject. Lynn Margulis was the first wife of Carl Sagan, and to put her in perspective, she was the formulator of the endosymbiotic theory of eukaryotic evolution. Boy, that's a mouthful. That's the theory that says that more complex eukaryotic cells arose in a single event when a archaeobacteria played host to a eubacteria, and that eubacteria, which was anaerobic, then settled down permanently inside the cell and became a mitochondria. Dawkins, Professor Richard Dawkins, described that theory as one of the great achievements of 20th century biology, which kind of puts Professor Margulis in perspective. Here are her own words. The oxygen holocaust was by far the greatest crisis the Earth has ever endured. Many kinds of microbes were immediately wiped out by the presence of oxygen. And this is the key sentence. Microbial life had no defense against this cataclysm. This is the accepted version of events. Evidence for it is not there. One of those educated guesses that plague evolutionary biology. Now, Christopher Hitchens, I don't know if you guys are familiar with him. He's dead now, but he was a very public intellectual and journalist, and also a very enthusiastic supporter of the Scottish distilling industry. He once said, an assertion made without evidence may be dismissed without evidence. But there is evidence that this was not the case. It's not entirely persuasive. It's not the sort of evidence that's absolutely conclusive, but it is persuasive, I think. And let's start by having a look at photosynthesis. As we all know, carbon dioxide plus water with the energy of the sun, catalyzed by chloroform, produces carbohydrates plus free oxygen. And the thing to note here is that respiration is simply photosynthesis in reverse, driven the other way by a different set of enzymes, predominantly something called ATP synthetase. The other thing to note is that whilst intuitively you might think that the oxygen released from photosynthesis comes from carbon dioxide, it actually comes from the water molecule. And we know this from isotope experiments. It would be much easier energetically to split the oxygen from carbon dioxide, but it's actually split from the water. All right, let's look at the evolutionary conundrum that is photosynthesis. What came first, the photosynthetic chicken or the oxygen defenses egg? We know that oxygen strips electrons from other molecules. We know that it shreds organic molecules, including DNA and RNA and proteins. At concentrations as low as 5 parts per thousand, it can initiate cell death. So how did cyanophytes survive their own toxic byproduct? How did these photosynthetic bacteria that evolve a biochemical pathway that results in the production of oxygen if that oxygen would absolutely be lethal to the cell producing it? Now, natural selection is a reaction to existing conditions. Life cannot anticipate future needs. So unless the proto-cyanophytes had some way of dealing with the presence of oxygen, photosynthesis would be maladaptive in the sense that it would kill whatever was trying to evolve it. Answer to the conundrum, interestingly, lies way back in the 1950s. This was when the world was being neurotic about this particular potential disaster, not this particular potential disaster. Two researchers from the University of Rochester, Rebecca Gershman and Daniel Gilbert, published a paper in the journal Science, and the name of the paper was Oxygen Poisoning and X-Irradiation, a Mechanism in Common. Okay, this is when we get to the tricky bit of biochemistry. We know the radiation poisoning is a four-step electron transfer process, and it's probably best understood by people who don't have a grasp of quantum physics like me, in the sense that energy, as we know, comes in discrete packages, which we call photons. And a single photon of energy can only interact with a single molecule or a single atom. One molecule of water, whether that's a photon of ultraviolet irradiation from the sun or a photon from a nuclear explosion, that water can only split into hydrogen and a single atom of oxygen, which is grossly unstable. And what we know happens, although we're not sure about the details because this reaction is virtually instantaneous, is that electrons are passed down a chain of intermediate products, a hydroxyl radical, hydrogen peroxide, and something called the superoxide radical, which is O2 minus, not O2 minus, O2 minus. In other words, one electron away from molecular oxygen. And it is these intermediate products which, in the presence of hydrogen atoms and hydrogen ions or protons, do the actual damage caused by irradiation. It's the hydroxyl ions that react with protons and DNA and RNA and shred them, and in doing so produce other radicals that carry out further damage, and you get these chain reactions which can affect up to 100,000 molecules. So it's a very destructive process. And there's also something called the Fenton reaction, where the superoxide radical, which is the last in the chain, reacts with ferric ions, which are common in proteins in the heme group. The heme group, for instance, which we have in our own blood in the form of hemoglobin. The critical thing about this, which the two researchers found, was that whilst water can be split down this chain of intermediates to form molecular oxygen, the action of molecular oxygen is exactly the reverse. And if you want an idea of how much energy is involved in this, just that iconic photograph might give you some idea. That's what happens when hydrogen and oxygen mix. So these intermediates do all the damage associated with both irradiation and oxygen poisoning, and they're known as free radicals. That's free radicals as in the multi-billion-dollar anti-aging industry, not free radicals of this particular type. So now we're getting to the heart of it. Oxidative stress is the presence of these free radicals. Radiation and hydrogen act identically at cell level to cause oxidative stress by producing the same free radicals. The reason they act on water is simply because it's so abundant in the cell. And of course, any defense evolved to deal with radiation would pre-adapt life to oxygen. So was the early Earth subjected to oxidative stress? Well, there are several theories about what the early atmosphere consisted of. There's the Oparin-Haldane hypothesis. They looked at the formation of the solar system. They said, well, it all arose from the same cloud as cosmic dust. Therefore, at the beginning, the atmospheres should all have been more or less the same. Excuse me. They analyzed the atmosphere of Saturn, discovered it from chromatography that it consisted of ammonia, methane, and hydrogen, and said, well, that's what the early Earth's atmosphere was like. That led to the famous Miller-Urey experiment, where you put that mixture of gas into a glass container, add an electric spark, and over a period of days come up with complex organic molecules. That's how we thought life originated. But if you look at the rocks, the ancient sedimentary rocks in Western Greenland, the fact that they're sedimentary rocks would suggest water vapor in the atmosphere in a hydrological cycle. The presence of nitrogen in our current atmosphere, there is no known process that could put it there, so we have to assume it was always there. And in those sedimentary rocks, those ancient sedimentary rocks, we have carbonates, which would suggest the presence of carbon dioxide. But either way, everybody seems to be agreed that there was no oxygen worth a damn in the atmosphere. About 1 part per million is the estimate. If there's no oxygen in the atmosphere, there's no ozone layer. If there's no ozone layer, the amount of solar ultraviolet radiation at the Earth's surface is about 30 to 40 times what it is at present. This guy, James Casting, worked for NASA. In the 80s, he was asked to do theoretical work on the production of hydrogen peroxide, one of those destructive intermediates in the splitting of water, or the action of oxygen, at the Earth's surface, assuming an atmosphere like today without the ozone layer. And what he determined was, in the presence of sunlight, without the ozone layer and the similar atmosphere, per square centimetre at the surface of the Earth, per second, 100,000 million molecules of hydrogen peroxide would be produced. That is a fearsome number until you calculate that that's 1/56 millionth of a gram of hydrogen peroxide. But it's also fairly clear that the surface of the Earth was indeed under oxidative stress. We just pause a minute to look at this particular issue. James Lovelock, the author of the Gaia hypothesis, has calculated that under present conditions, with an ozone layer, with the action of sunlight on water, the amount of hydrogen and oxygen produced is equivalent to the loss of 3 million tons of water per annum. It sounds an awful lot, but he's also calculated that that would take 4.5 billion years to reduce the planet's water by 1%. So there's obviously a lot of water on this planet. And what happens is when a molecule of water is split, the hydrogen ions float off into space because they're too light to be contained by the Earth's gravity. The oxygen goes into the atmosphere. When you've got oxygen in in the atmosphere, or enough oxygen in the atmosphere, the chances are that those hydrogen molecules on their way out into space bump into an oxygen molecule and form a molecule of water, which then goes back into circulation. And that's how oxygen in the atmosphere preserved the oceans. So basically we have this thin green pond scum, these chains of cyanophytes, to thank for the fact that the surface of our Earth doesn't look like the surface of Mars. Other evidence along this line is an enzyme by the name of catalase. Catalase is found in all forms of aerobic life and most forms of anaerobic life, including anaerobic bacteria. And it's incredibly efficient at mopping up hydrogen peroxide. Now, if we go back to the photosynthesis equation, we know that in splitting that water molecule to form glucose, electrons pass down a chain of intermediates. And those electrons can do an awful lot of damage. And that damage is presented in the cell because photosynthesis takes place within a molecular cage, which is known as the oxygen evolving complex. And what's fascinating is that the oxygen evolving complex is almost certainly two molecules of catalase joined together. And if that is true, then it would also infer that this antioxidant enzyme was around before photosynthesis took place. Also, probably interesting to point out that bacteria have other defenses against radiation and oxygen poisoning. They hide in anaerobic and anoxic mud. They hide behind layers of dead cells. If you look at stromatolites, underneath the layer of photosynthesizing cyanophytes, you have a layer of dead cells, and underneath the layer of dead cells are colonies of anaerobic bacteria. They buffer. That's what happens here is, for instance, with sulfate-reducing bacteria, they reduce the sulfate, they release sulfur dioxide. The sulfur dioxide leaks out of the cell. If it comes into contact with an oxygen molecule, it reacts, but it reacts outside the cell at a safe distance, like exploding a bomb far away from you. And that reaction produces another sulfate group, which the bacteria can then reprocess. They also move. We know that bacteria can move down an oxygen gradient from high to low using their flagella, and they use mucus. Mucus we generally associate with a blocked nose, but mucus consists of chains of polypeptide molecules, it's a polymer, and each one of those polypeptide molecules carries a negative charge, and what happens is that this coating of mucus around the bacterial cell, carrying a molecular negative charge, attracts positively charged metal ions. And whenever an oxygen molecule comes anywhere near those positively charged metal ions, the oxygen reacts with the metal to form a metal oxide, and thus protects the bacterial cell. Apparently, at the pre-extrual water purifying works in Hermanus, they use this type of bacteria to extract manganese from the groundwater. So, in summary, because the early Earth was under high levels of solar ultraviolet radiation, unfiltered by an ozone layer, Life had to be able to cope with oxidative stress through antioxidant enzymes such as catalase, and of course defenses such as mucus. This pre-adapted proterozoic life to the evolution of photosynthesis, as these antioxidant defenses were defenses against ultraviolet radiation as well as defenses against oxygen. Oops, I think we've gone over.
Keep going.
Don't worry, they've given us, and they've taken away the limit. You can carry on.
Okay, there we go. So, being pre-adapted to oxygen poisoning, anaerobes probably most likely didn't die out wholesale as the great oxidation event orthodoxy supposes, and it was more likely a period of rapid evolutionary change rather than a mass extinction. The consequences longer term, oxygenic photosynthesis by billions of cyanophyte bacteria changed the planet in utterly profound ways. Free oxygen preserved the oceans, as we've examined. It changed the chemistry of the atmosphere, the lithosphere, and the hydrosphere. It created an environment for the evolution of aerobic respiration, which was simply photosynthesis in reverse. And aerobic respiration makes available a five-fold increase in available energy over even the most efficient forms of anaerobic respiration. Aerobic respiration impacted on the carbon cycle. It possibly saved the planet from a carbon dioxide famine because aerobic respiration replaces carbon dioxide instantaneously back into the atmosphere. It led to an increase in size and cellular complexity and then multicellular complexity of the eukaryotes. And as a response to the use of oxygen in respiration, we had to invent sex and mortality, but that's a story for a different day. That's the end.
Wow.
Well done.
All right. I'll try and answer any questions.
Well done, Ian. Good start to the process, and we'll learn from it.
No, that was excellent. Much of it I may not have understand entirely, but it certainly puts a lot of thoughts on early life into some perspective.
Okay.
Possible to get a copy of some of the slides. There are one or two there that I think adequately describe my friends, and I need to send it on to them, particularly that one of the right near the beginning of microbes.
Yes, I've forwarded a copy to John. I don't know if you got it, John.
Yeah. I mean, I agree, Gordon, that we should actually. I don't think any of this, Ian, will have no problem circulating it. It's a good presentation put together from public domain stuff, and it'll be. So if you're happy, Ian, we'll circulate it to everyone.
Yeah, with the greatest of pleasure. And look, if anybody wants to borrow the book, I'm more than happy to lend it.
Yeah, and if people have got questions, they can come back to you as well, Ian.
And
Okay.
on top of that, I've recorded this session so you can actually listen, or somebody who's missed it can listen to the presentation and watch the slideshow as well.
Well done, Henny.
We'll put up. I'll give you a link.
Okay, comments from everyone. I mean,
because I know a lot of the guys that actually tried to get in, like this morning, who didn't get in, but I think this is a very good first try. Thank you very much.
Very nice. Yeah.
Thank you.
Yeah, I think it's excellent. Thanks for everyone. Thanks, Ian. And we should discuss, Henny, that maybe if people are happy while we lock down, we've got a bit of spare time. We could say do it fortnightly. or more often if people have got presentations ready, and then we should set them as 20-minute presentations, max
Yeah.
25, with 5-10 minutes for discussion.
Yeah.
I think that's an
I
excellent
saw-
idea.
Sorry. Gordon?
I think it's an excellent idea. When we finished talking about the presentation, I picked up a few tips on working with Zoom that I think other people might benefit from.
Yeah, absolutely, Gordon. Have you got some comments, or are you going to circulate it? Do you want to talk?
Yeah, look, the trick is everyone other than the presenter mutes their audio and switches off the video. When you want to make a comment, you touch and hold down the space bar on your computer or whatever you're using, and it unmutes you momentarily until you release it. That's a great trick. Gets rid of the background.
Yes, so you don't have to to activate your mic. Also, the presenter can mute everybody. And also in the middle, when you see participants, when you click on participants, you see there's a little hand icon. If we get to that discipline where you put your hand up, then the presenter can say, okay, John, I see your hands up. What is your question?
Okay, any other comments?
Maybe just one from me. The GSSA is
Craig,
discussing
hi.
doing. Can you hear me?
Is that Craig?
Yeah, go for it. Craig Smith. Yeah.
The GSSA is about to send out a call for presentations of this nature to do the similar thing. I'm glad to see that this worked very well. I presume the license that you have is for under 100 people?
Yes, it's a free version, and as we ran out of time, there was a little message on my screen that says, "There's a present from Zoom. We're giving you free time to finish your talk."
Yeah,
Okay.
Craig. So this is so this works for 40 minutes. That's why, and that's why we should do like 20-25-minute presentations. Obviously, it takes a few minutes to get in, do the presentation, and then have some time for questioning. Again, it's just good discipline. And I think all of us.
Another option is to do it through the GSSA has actually purchased a 100-person license with no time limit.
Okay.
It's not that expensive. It's going to work out
What
about
does it cost?
2,000 rands a year or something like that. But if you wanted to run a meeting using the GSSA license, we can organize that.
Okay.
That's great, Dan.
All right.
Now you're talking. Thank you.
Yeah. Yeah, but we should still have a discipline that the talk doesn't go too long, not to be disrespectful to anyone, because the more important part is actually the interaction and discussion. And if people got questions, we want to talk about it. So we should have more short presentations and some good discussions. That's my suggestion.
Okay.
Okay, but anyway, please send your comments back to Henny and myself on the email, or circulate them. We're all learning. It was a great start. And thanks, Henny, for